Today, Dr. Cothran will speak on DNA, past, present, and future. We will be unable to take questions during the webinar, but invite you to submit your questions to the website, chsa@caspian.org, and we will be happy to pass them on to Dr. Cothran for a reply at a later date. And with that, I hand the webinar over to you, Dr. Cothran.
Thank you very much. I'm not quite sure exactly what is expected here. I was actually kind of anticipating answering questions as they came, but let me just kind of give you a little history of genetic testing of Caspians and what we do in terms of genetic testing, the different kinds of genetic testing that we can do, and then we'll go from there. Maybe Tina or somebody else can ask some questions on things that I've missed. So just a little history of my own involvement with horses and genetics of horses. I started my career working on wild populations of rodents predominantly, and then kind of moved into working with white-tailed deer, doing genetic testing, genetic management. And I guess about 1982, I was looking to move on from the position I was in. It was not a career position. It was a temporary sort of thing. And I ended up getting a postdoc at a place called the Southwest Foundation for Biomedical Research in San Antonio. And the project that I was hired to work on involved genetic variability and reproductive success in standard bred horses. So that was my introduction to the genetics of horses. After about 3.5 years at the foundation, where I continued to work on the standard bred problem the entire time, although I did other things as well, I moved to the University of Kentucky because I was looking to get to a university. And there I worked in the biochemical genetics section of the equine parentage testing laboratory at Kentucky. And our primary function was doing genetic testing of various horse breeds for parentage verification. And at that time, the genetic testing we did was based on what we call blood typing, which includes 2 different types of genetic markers, blood groups that are very similar to the ABO, RH type of blood groups loci in humans. So my simple blood type for that is O positive. The horse blood types that we worked with were a bit more complicated than that, at least most of them were. But we also did biochemical genetics, which is what I had done pretty much from the beginning of my professional career. So what that entails is we need a blood sample of a foal and its presumptive parents, its sire and its dam. And we test them for the marker systems we can test, and we compare the results, the genotypes, at each of the systems that we get, and we look for compatibility. So basically, if the foal has a particular characteristic at its phenotype for one of the systems, then the parents must have the same factors at their type in such a way that is compatible with basic genetic inheritance. And we do that with the blood typing. We looked at 17 genetic systems, some of which were quite low variability, not very powerful, but others were quite high. And so we could do a parentage with what we call an efficacy of about 96 to 99%. And efficacy really refers to the probability of being able to detect an incorrect parentage. So that's what we're really doing. The process is not confirming parentage, it's not proving parentage, it's showing that there is no incompatibility or exclusion of parentage. And so, I don't know, some of you may have gotten blood typing reports or seen blood typing reports from my laboratory there, and the parentage result, if it qualified, was we reported it as parentage not excluded, because that's actually what we were doing, is trying to exclude, not trying to confirm. So that's an important thing to understand. However, because the systems are so powerful, if we get a not excluded, that is very close to being confirmation and proof of parentage. It's just not scientifically exactly proof. Well, somewhere around 2000, the year 2000, we moved from blood typing to DNA testing. So blood typing, we're testing parentage, proteins, which are gene products. And they were in fact true gene products from a protein coding gene. But when we moved to DNA testing, we started testing a kind of genetic marker system called a microsatellite. And this is a place in the DNA where a very short segment of the building blocks of DNA, the nucleotide bases, are repeated a number of times. And the variability is in the number of times that little repeat is repeated. And these are extremely variable systems. And that's because of the nature of how the variation occurs, this repeating sequence, that's subject to error during DNA replication, cell division, the production of gametes, so that that increases the variability over a population over time. And so they're extremely powerful. And so we moved our efficacy up to 99.99% or even higher in many cases, depending on the variability of the breed. Well, sometime in the really the late 1980s, early 1990s, I started getting interested in testing various breeds of horses to try to understand the relationships among breeds, the degree of genetic variation in breeds. And so I started looking for sources of samples from the various breeds. And one of the people that helped me a great deal was a writer who was writing at that time the International Encyclopedia of Horse Breeds, I believe that's the name, Bonnie Hendricks. And she was very interested in what I was doing. So she was instrumental in getting me in touch with the various people who had access to different kinds of breeds. And one of the ones that was especially interesting to me at the time was the Caspian. And the reason for that is some of the early work that suggested that it might be ancestral to the Arabian type horses. And so that was interesting. And I wanted to see what I could tell in that regard through my genetic testing. And we were doing blood typing when we first started. And I got in touch with Louis Ferrous via regular postal mail, which is the only way we had to do it back then, until later we got fax numbers that we could exchange. And we corresponded, and through Louise, I got in touch with the Caspian Society in England and Brenda Dalton, and we started getting samples of the horses and started doing blood typing of the Caspians in Great Britain. And through time, we started doing parentage verification for many of those, although I don't remember exactly the way the sequence worked back then. But of course, at the Animal Health Trust, they also have a horse parentage testing lab, and I believe a lot of the testing moved there. I could be wrong on that. We did a lot of it, I remember for sure. So in around 2000, we started converting to DNA. We started converting the database we had. You can't convert a blood type into a DNA type just simply by looking at a table and changing this one system. It's completely different systems. So we had to retype any animal that had been blood typed and that we needed to do for parentage verification. We had to retype them using the DNA markers, which we did. And so somewhere around 2000, we started doing DNA typing for Caspians and all the other breeds. And over the course of time, I managed to get samples. I guess somewhere right now, I have DNA data for over 150 different breeds of horses from all over the world. Certain big holes, very, very few samples have ever been tested from China. In fact, I don't think I have any breed from China that is actually fully DNA typed. But anyway, that's helped us in our analysis of genetic relationships among horse breeds. And because of Louise, I managed to get a number of different breed samples from Iran, including the Turkoman, the Kurd, we got Persian, Arabian, and more recently, we've gotten a couple of other different types of Iranian horse breeds. So we've been able to look at the Caspian and how it relates to other Iranian horses. And we've also then compared the Caspian to horses from all over, really all of the horses that we have in the database. We also started doing DNA testing of Caspians for the registries in the US. And we also did some for some of the other international registries. We did some from Norway, we did some from Sweden, and a little bit of others, but mostly those places moved over to their national laboratories. Virtually all of the major countries that are likely to have a large horse population, including something like the Caspian, are going to have their own national laboratory, often associated with a university as ours are, but not always. So at this point, the vast majority of Caspian horses have been DNA typed, and that's going to ensure that the pedigree of these horses is highly accurate. Now, once we started developing the DNA technologies, we also started developing the ability to test for certain phenotypic characteristics of horses. Coat color was one of the first things that became available. There are a number, in fact, quite a large number of coat color genes that have been identified and we're able to test for them. Not something I see done in Caspians very often, but it's possible. We developed tests for a number of genetic disease genes. So we are now able to test many types of horses to see if they carry a particular genetic disease, which can aid in avoiding having the disease occur in a population or in the foals. A few other characteristics. And then just about 5-6 years ago, really, I guess it actually goes back a little bit further, but the technology became available for doing whole genome sequencing of horses at an economical cost. It's not the kind of thing we're going to do for individual horses for something like parentage verification right now. It's still much too expensive for that, but it is something we can do for looking for genetic characteristics or for doing population studies, evolutionary relationships of horse breeds to each other and that sort of stuff. And a lot of that is underway. It also would allow us to basically have the genetic blueprint of any particular horse breed. And that's something we've talked about. We haven't really implemented it yet just because of the logistics. The cost of the DNA sequencing has gone way, way down. However, the actual total cost is still pretty high, mainly because you have to employ somebody to analyze the DNA sequence. And that requires paying salaries, which is a lot more than paying laboratory cost. Let's see, where should I go? The work that I have done in terms of relationships does show that the Caspian fits within the group of horses that includes the classic Arab breeds. Other breeds such as the Turkoman and Kurd are in that group, the Akhal-Teke. The Caspian does occur in certain combinations of the breeds that we analyze. This is a complex subject and I'm not going to go into detail on it right now. But it does occur in a position that is basal to the cluster of breeds in a tree of relationship that suggests that the Caspian could be ancestral. So there is some genetic evidence of the possible ancestral relationship of a Caspian type horse to the Arabian breed, but we really need to examine that in far more detail and probably using whole genome sequences to really work that out. We can say there's suggestion that it could be ancestral, but I think that's as far as we want to go right now. Let me just take a quick look here to some of the topics that I had been sent as suggestions of things to work with, and we'll see if what I'm forgetting. Certainly, okay, one thing is, how is the testing done? So blood typing was quite complex in the laboratory. The blood group testing involved two different types of tests. One where we, in general, blood typing involves exposing blood cells to antibodies against the various factors that are inherited. And that either causes the red blood cells to burst because the antibodies destroy the factors on the surface of the red blood cells and cause the membranes to burst, called that hemolysis. The other is it causes the antigens on the surface of the red blood cells to stick to each other, so the blood clots, essentially. We call that agglutination. The biochemical genetics required about 5 different genetic test systems to do the 10 to 11 genetic loci that we typically did testing there. So it's quite complex. It required producing antibodies, which for the laboratories that did a lot of testing and tested a lot of different breeds, that pretty much meant that you had to maintain a herd of horses. At the University of Kentucky, we maintained a herd of about 80 horses. So all of you people who are breeding horses, you understand what an expense that entails. The personnel involved and all that sort of thing to keep 80 horses healthy, and also to do all the things that we needed to do to produce antibodies. DNA testing came along and that changed everything. Although the DNA testing is a fairly complex biochemical process, compared to the blood typing, it is really quite simple. The number one thing that drove the change from blood typing to DNA typing was that you could use hair follicles to test the DNA systems, and it did not require bleeding a horse to get a blood sample and then rushing the blood sample to the laboratory so it didn't spoil before it could be tested. That was particularly difficult for international exchange of materials. What we simply do is we get the the hair follicles, clip them off into a little well in a tighter dish, and add some enzymes which basically digest the protein in there. We extract the DNA, we subject it to the polymerase chain reaction, which amplifies the DNA from a tiny amount to as much as we want to make, pretty much. And in the process of doing that, we attach marker system labels, fluorescent dyes, to the amplified DNA so that we can detect the specific pieces of DNA in the entire genome that represent the different microsatellite systems that we use for parentage or research testing. These are then subjected to a process called electrophoresis, which is simply putting the labeled samples into a porous matrix, running electric current through it. The DNA migrates in response to the electric current, and the distance it migrates in a given period of time depends on the size of the fragment of DNA that we have amplified. And so we can actually, with the equipment that we use, they're called sequencing machines. They were really designed to do whole sequencing, but they can do what we call now fragment analysis of the microsatellite DNA fragments. And there's a laser in the sequencing machine that when the DNA fragment passes through a little window where the laser is going back and forth like that, really, really rapidly, the tags on the fragment for each of the systems will fluoresce, and they're different colors, red, blue, and green, and yellow. The red is actually used as a size standard. And that flash of light is recorded and sent to the computer, and compared to a size standard in the gel, it identifies the size of the fragment. And so it's really a very simple process compared to the blood typing. And we can test, in our system, we test 8 systems in one run and another 5 systems in the other to get the 13 systems we use in routine parentage analysis. It's again the genotypes or the phenotypes are actually recorded by the computer and sent directly into the computer. It does not necessarily require human scoring of the results, although we do check every result by eye to make sure that the computer is calling it the way it's supposed to be called. Now, again, if you have seen a DNA typing report, you'll see a system of undecipherable letters in the DNA type. And what those represent are the names of the microsatellite systems and a code for the variants that occur in each of those systems. So on our types, the very first of the microsatellite systems that is listed is VHL20. That's V as in Victor. And we might have a type that is LM. The systems themselves are named using abbreviations for the laboratory that discovered that particular system. So VHL stands for Van Heringen Laboratory, which is the Dutch laboratory. The letters for the particular variants are based upon a standardized, nomenclature system that was developed by the International Society for Animal Genetics, which almost all of the laboratories are members of. And the way it works is we measure the molecular size of each of the variant fragments for each of the systems. And they might range from, say, 100 nucleotide base pairs to 200 nucleotide base pairs. And we take the one that is in the middle, say 150 base pairs, and give it the name of M. And then every one that is stepwise larger or smaller, we go up or down the alphabet. So the next molecular size heavier than M would be N, and the one lighter than M would be L. And so it's just a simple system of nomenclatures that is standardized among all laboratories around the world that do horse parentage testing utilizing the ISAG, International Society for Animal Genetics system. And so we're very freely able and confidently able to exchange types among laboratories because they've all undergone a system of comparison tests to make sure that they are doing it right and that provides the results to all the other laboratories so that everyone can have confidence that every other laboratory is doing the same thing. Okay, so that's kind of the practical aspects of DNA typing. The other thing that we can actually use the DNA typing results that we get from the parentage testing for are population studies, studies of genetic variability, where we can compare the variation at one breed to that in another breed. And this can give us an idea of the genetic health of the population in terms of variability. That, in essence, is a measure of inbreeding. Inbreeding is more effectively and accurately calculated using the pedigrees, but you can use the variability measures to get good estimates of inbreeding. So that's a very valuable way to do things. And computationally, it's a lot simpler than using the